Morning students, yesterday we have completed the topic amino acids and their classification. Today I am going to deliver another topic, that is structure of biomolecules, that is proteins, of unit 3rd for class 11. So today we will learn what is protein and how protein is made up of. So students, first of all I will ask you to write two simple amino acids. So one is glycine and another I will taking alanine. So now I will ask her to draw their structure formula. So COOH, CH2, NH2, here NH2, CH. See, I am writing amino acid on the left hand side because most of the amino acids are on left hand side rather than right hand side. So this is the structure of alanine and this is the structure of glycine. So students, whenever an amine NH2 and COOH are closer to each other and you will heat there, then there is a OH from COH group, carboxylic group, and H from amine group will release H2O, water molecule, and form a new bond, that is NH2 CH2 C double bond O NH CH CH3 COOH. Students, this new bond is known as, this new bond CONH is known as a peptide bond. A peptide bond. So students, how a peptide bond is formed? Peptide bond is formed by the mixture of COOH group and NH group. So how many amino acids I have taken? Students, I have taken two amino acids. That is, one is glycine, in short I will write GLY. Another I will take alanine, in short I will write ALA. So two amino acids joined to form a bond that is known as dipeptide bond. When three amino acids are joined together, they make bond that is known as tripeptide bond. If four amino acids are grouped together, joined together and make a new bond, then tetra and so on. Students, if the number of amino acids is more than 10, then means amino acid, that mass of amino acid is known as polypeptide. So polypeptide is formed by the number of amino acids. So obviously its mass is very high because it is made up of number of amino acids. So if the molecular mass of a polypeptide is more than 10,000, that is 10 raised to power 4 AMU, atomic mass unit, then that polypeptide is known as PROTEIN, protein, which is our topic. So students, how protein is formed? When many amino acids joined together, they make a polypeptide, and when the mass of molecular mass of one amino acid is more than 10,000, that polypeptide is known as a protein. So this is about the formation of protein. Now I will write some classification of protein. Classification of protein. Proteins are classified on two bases. One is fibrous protein, another is globular protein. Students, fibrous proteins, as for the name suggests, these are fiber-like structures. Means they have long strands They have long strands. These long strands are made up of polypeptide. So they run parallel to each other. They run parallel to each other. On the other hand, globular proteins are the polypeptide chain. The polypeptide chain, it coils. If it is polypeptide, it coils like spring-like structure. It coils and forms a spherical globule. Spherical globule. So it forms this like structure, spherical globule. Such type of proteins are known as globular proteins. So here keratin, which is the fibrous protein, mainly present in, which is present in our hairs, which is present in wools, which is present in silk. So students, do our hair soluble in water? No, it means fibrous proteins are insoluble in water. Insoluble in water, whereas these are soluble in water. So here two examples are albumin, insulin are the examples of globular proteins. So these are all about the classification of proteins. Now I will describe what is the structure of protein. Structure of protein. To study the structure of protein, I will classify it into four main parts. First part is that primary proteins. Primary proteins. Students, primary proteins are basically the sequence of amino acids. What are these? These are sequence of amino acids. So, for example, I will write glycine, alanine, leucine. Glycine, leucine, alanine. Here the tripeptide amino acids, both the contains tripeptide amino acids. If I have changed their sequence, and I changed the sequence, their polypeptide will be different. So in short, primary amino acids are basically the sequence of basically the sequence of amino acids in a polypeptide chain. Secondary structure of protein. Secondary structure of protein. Here, to study the secondary structure of protein, either it is in the form of alpha helix, or it is in the form of beta pleated sheath. Beta pleated sheath. So, if I have got a long polypeptide chain, this is polypeptide. If I have a spiral-like structure, then this is known as alpha helix. This is known as alpha helix. If it is in the form of this like structure, layer-like structures, these polypeptides stretch. They stretch to maximum extent. So these layers which stretch to maximum extent and forms a sheath-like structure. This sheath is known as beta pleated sheath. Students, what helps the polypeptide chain coils around itself? How it is going to coiling around itself? We have already learned that there is a carbon double bond NH, C double bond O, amine NH. There is a hydrogen bonding takes place. So due to this hydrogen bonding, they are coiling around itself. So in alpha structure, hydrogen bonding takes place, whereas in beta sheath structure, here is also I will write R CH C double bond O NH, R CH C double bond O NH, or CH C double bond O NH here, or CH NH C double bond O, or CH NH C double bond O, or CH NH C double bond O. How these beta places are connected to each other, or how they are attracted to each other. These are different amino acids forming polypeptide chain. I have kept a polypeptide chain parallel to each other. You can make hydrogen bonding. In this way. They are parallel to each other, and they can occupy maximum space also. So this is about the secondary structure of protein. Now comes to the tertiary structure of protein. Tertiary structure of protein. Here the how the overall folding of protein occurs in 3 dimensions. For example, I will make a 3-dimension protein, polypeptide protein in 3 dimensions. So this overall arrangement of folding of proteins in 3 dimensions is known as tertiary structures of protein. Students, what are the forces of attraction which are responsible for the secondary and tertiary folding of proteins? So first of all, we know one is clear, that is there is a hydrogen bonding. First force of attraction is that there is a hydrogen bonding. Second is that there is a van der Waals force. There might be wall to wall force, that is known as van der Waals force. And another is the important force is known as disulfide force linkage, that is SS linkage. SS linkage means most of the amino acids contains sulfur also. So for example, methionine and cysteine, etc. So there is SS linkage, disulfide force of attraction. And another important force is electrostatic force. This electrostatic force is mostly seen in Zwitter ions. For example, NH3CHCO minus. So this is charged species. So these are known as electrostatic force. These forces are responsible for the secondary and tertiary coiling of proteins. So this is about the tertiary structure of protein. Now coming to the quaternary structure of proteins. This is the fourth structure. Certain proteins, or if a protein is if a protein is made up of more than one polypeptide chain, such structure is known as coordinate structure. For example, if it is polypeptide first, second is just like this is polypeptide second, third is just like this is polypeptide third. How these polypeptide chains link it to each other, or how they are polypeptide chains joined together with respect to each other is known as subunit. What it is known as subunits. And whole n together constitutes a protein. And the structure is known as quaternary structure of proteins. Students, we will discuss denaturation of proteins in